Antibiotic-resistant bacteria are on the rise, and it's imperative that new drugs be developed to fight them. Hi, my name is Kwong, and I'm a Beckman Scholar in the Shell Lab at UC Davis. As a pharmaceutical chemistry major, I'm interested in using synthetic chemistry to further studies on unexploited drug targets. Currently, we are met with the challenge of bacteria becoming rapidly resistant to available drugs. In addition, there's less research being done into new antibiotics due to scientific difficulty and profit margin considerations. This unfavorable combination has led to the creation of untreatable bacteria like MRSA and less options to treat infections. What's even more concerning is that some gram-positive bacteria are becoming resistant. These bacteria, compared to the gram-negative bacteria, are traditionally easier to kill. This makes it even more important to conduct research in this field of study. Antibiotics normally target one of three essential bacterial processes, such as cell wall synthesis. Our target protein is involved in nucleic acid synthesis. My research involves the synthesis of small molecules, called gyramides, that inhibit DNA gyrase. DNA gyrase is involved in DNA replication. When the DNA helix is unwound, it causes a tight raveling known as supercoiling. DNA gyrase is involved in releasing supercoiling by cutting and reattaching the DNA strands. If this enzyme were to be inhibited, we would be left with damaged DNA, killing the bacteria and stopping any further growth. Furthermore, DNA gyrase is an enzyme that's specific to bacteria. This makes DNA gyrase an excellent antibiotic target since it won't hit human proteins. Right now, drugs in the quinolone class, such as ciprofloxacin, target DNA gyrase. Unfortunately, these drugs are becoming resistant to these drugs as well. However, we believe that our gyramides have a different binding site, as well as different resistant determining genes. Therefore, quinolone-resistant bacteria will be susceptible to gyramides. This is an example of a gyramide. A typical gyramide is made up of three parts: a sulfonyl group, a pyrrolidine ring, and some sort of substituted benzene. Let's just call these regions A, B, and C. In order to find a gyramide that can best fit the binding site, we can swap out either A, B, or C for various combinations. These combinations can be put into a library and sent through a high-throughput screen to see which compounds would be most likely to perform the best. Part of my research involves synthesizing select compounds in greater quantities so that collaborators can run toxicology and pharmacokinetics tests. Shown here are the structures of the analogs that I synthesized from the library. As I explained earlier, you can see that what we called regions A and C are changing to make different analogs. You might be wondering why there are no variations to region B. Previous research has shown that this particular orientation of the molecule, or in chemistry terms, stereoisomer, has better activity. That particular isomer is indicated by the bolded line, which shows that region A is angled slightly upward. We deduce that having region A angled upward fits better into the binding pocket, compared to it being angled back. Another way to potentially develop a better fitting gyramide is to look at the approximate shape of the binding site. One way to do this is to make photoaffinity reagents. Photoaffinity reagents are deliberately designed so that one part of the molecule binds into the binding site, and the other part forms a reactive species when treated with light. First, DNA gyrase or the bacteria is treated with the small molecule. It's then treated with UV light, and the reactive species will covalently bond with any nearby amino acid. From there, the protein can be digested and sent into a liquid chromatography mass spectrometry instrument, which will separate the fragments and detect any extra mass from the photoaffinity reagents. We can then deduce which amino acids are in or nearby the binding site by using the sequence of the protein, and further tailor the gyramide structure to the binding site. In order to determine if our molecules are getting into the bacteria, we can run a simple biological assay known as minimum inhibitory concentration, or MIC. This is a technique used to determine the lowest concentration needed to kill bacteria. A small amount of drug is dissolved into solution, and then diluted in a series of concentrations for testing. Desirable MICs would be in the single-digit micromolar range. Shown here are the pictures of the MIC assays done by the Weibel group at the University of Wisconsin-Madison for 2 of the photoaffinity reagents. While the MICs aren't great, it shows that our drug is still getting into the bacteria, and photolabeling can proceed. Remember how I mentioned that region B isn't changed at all between analogs within the library? Well, ongoing work involves making new analogs outside the ones in the library matrix. We are testing the effects of having a primary amine on the molecule, and one of these analogs alters region B. For future work, we would like to keep exploring new analogs and design new molecules with the help of the photoaffinity labeling. I would like to thank Dr. Shaw and the Shaw Lab for mentoring me through my research. Special thanks to the Weibel group for doing all the biological assays, and a big thank you to the Arnold and Mabel Beckman Foundation for supporting this research.